Hi, I'm Payton,
And I'm Leah.
and we are going to be diving into the tardigrade phylum today. So tardigrades are a very specific group of organisms and therefore have many different distinguishing characteristics. Their bodies are short, plump, translucent, and covered in a tough cuticle that they have to shed in order to grow. They have five body sections, of which are a defined head and four body segments, each with a pair of legs. And tardigrades actually have no bones. They instead have a hydrostatic skeleton, which is a fluid-filled compartment known as a hydrolymph. Their spinal cord is much different from ours in that it's a ventral nervous system that sends signals between their brain and body. Their body cavity is an open hemicule that touches every cell, which means there's no need for a circulatory or respiratory system. And tardigrades have a full alimentary canal and digestive system, as well as a dorsal brain. Tardigrades are very, very small. They're only averaging about 500 micrometers in length, and they have four pairs of legs and four to six claws or sucking discs on each foot. The hind legs are actually attached backwards and are used for grasping and other maneuvers. They have a specialized mouth part called a bucopharyngeal apparatus, which allows them to actually suck the nutrients out of the plants and microorganisms that they consume. As for reproduction, some tardigrades are able to reproduce asexually and some produce sexually. Tardigrades can survive in extremely harsh conditions and therefore be found in about every habitat on Earth imaginable. They have been found in temperatures at absolute zero and high temperatures of 150 degrees Celsius. They can survive pressures of up to about 40,000 kilopascals, radiation 100 times the lethal human dose, and the vacuum of space. My favorite thing about tardigrades is they have this cool survival mechanism called tun state. Tardigrades, being naturally aquatic animals, need a film of water surrounding their bodies to allow them to perform cellular respiration. And without this, they dry up, curl up, and turn into this desiccated form called a tun. During this, they completely slow down their metabolism to less than 0.01% of normal and can remain in this state for over 30 years. Once rehydrated, they can bounce back to their original state and metabolism rate within minutes. Important subgroups. Tardigrade subgroups are distinguished by their physiological features and as well as the habitats where they reside, head size, segmentation shape and number, and the number of claws on each toe. The Heterotardigrada subgroup comprises of the orders Arthrotardigrada and Echinskoidea and consists of marine, freshwater, and semi-terrestrial species. The species in this subgroup tend to lack the cephalic sensory organs that serve as sensory receptors. They have legs with 4 to 6 digits with complex claws or discs, and their cuticles are armored with plates. The Eutardigrada subgroup consists of the orders Apocola and Paracella and consists of terrestrial and freshwater species. This subgroup is characterized by having cloaca, which combines the reproductive vent and anus. They have a straight midgut, a Malpighian tubal system, and two double claws on each legs, but they don't have any digits. And Eutardigrada also have a naked or smooth cuticle without any plates. The final subgroup, Mesotardigrada, is only composed of one species, the Thermozodium isaki, which can now no longer be found after its habitat, which is a hot spring in Japan, was destroyed in an earthquake in 2018. Because of this, only some biologists even recognize this group as a subgroup of tardigrada. Evolutionary history. Tardigrada were originally discovered by a German zoologist Johann Goes in 1773, calling them Kleiner Wasserbar or Little Water Bear. It wasn't until 5 years later that Lazzaro Spallanzani, an Italian scientist, named them Tardigrada or slow steppers after their sluggish speed. The group was officially recognized as a phylum in 1962. Now, molecular studies with 18S rRNA indicate that tardigrada are a monophyletic sister group of the arthropods. Morphological analysis with fossil lobopods and arthropods suggests that tardigrada branch between the Onychophoria and the Earthropoda phylums. It is known that tardigrada originated from the Lopopodians, which were soft-bodied worms with lopodious limbs, of which thrived during the Cambrian period. The marine heterotardigrada under the order Arthrotardigrada have the greatest number of plesiomorphic characters and are thus considered the sister group for all other tardigrades. Within the eutardigrades, the order Apocola is considered to be the sister group for all other eutardigrades. Fossil evidence. Due to the lack of any hard body parts, the fossil record of tardigrada is extremely scarce and obstructs the understanding of their early evolution. The fossil tardigrades, Milnesium swillinski and Paradorphorabirus chronocarabius, are the only ones with resolved taxonomic positions, which restricts the availability of calibration points for estimating the origin of this phylum. The picture to the right shows Canadian Cretaceous aged amber, in which two crown group tardigrades were originally found using confocal fluorescent microscopy. Formal morphological redescription of the first group, born leggy, an extinct genus of tardigrades, reveals its feature of Hypsibius-type claws, and Aerobius dactylus, the second group, was described by its unique combination of claw features. Now, phylogenetic analysis indicates that both of these genuses belong to the eutardigrade superfamily Hypsiboidea, which adds a critical calibration point. Tardigrades are also estimated to have early Paleozoic diversification. Evidence has gathered that independent terrestrialization of eutardigrades occurred around the end Carboniferous period, and terrestrialization for the heterotardigrada occurred during the lower Jurassic period.
Moving on to ecological significance. Tardigrades are ecologically significant because they play multiple roles in the ecosystem and they are able to survive in extreme environments. First, tardigrades are consumers at different trophic levels in the ecosystem. They act as herbivores, which feed on plants, omnivores, which eat food both from plant and animal origin, and carnivores, which feed on other animals. With all this being said, these organisms mainly eat plants and algae, bacteria, small invertebrates, and other tardigrades. Second, they are a pioneer species when they move into new environments. When they do this, they attract other predators and other invertebrates, which are cold-blooded animals with no backbone. Next, tardigrades are an important part of soil fauna. Soil fauna are organisms that live in soil or at the soil litter interference for a period of their life cycle. In addition, insects and earthworms chur the soil and promote the formation of soil structure, nutrient cycling, etc. Continuing on, tardigrades are extremophiles, which means that they can survive in environments that most other animals can't. They can also survive in extreme temperatures, high pressures, the vacuum of space, and up to 30 years without food or water. Next, tardigrades can enter a state of inactivity called cryptobiosis when conditions are not good. During this state, they squeeze out all the water out of their body, retract their limbs and heads, and roll into a ball. Finally, tardigrades can live in almost every habitat on Earth. This species can live from the top of the Himalayan mountain range to the bottom of the sea, to hot springs, and to icy areas. Many organisms eat tardigrades. Amoebas prey on tardigrades. Other tardigrades, mites, spiders, and insects eat them. Moving on to the topics of interest, I will be talking about three: survival, current status, and potential medical uses. Tardigrades are microscopic organisms that have survived on Earth for over 600 million years. Their unique abilities to survive in extreme conditions have made them a subject of scientific study with potential application for human health and agriculture. For example, tardigrades can produce proteins and sugars that help preserve biological materials like blood cells and vaccines. Tardigrade proteins and sugars could help improve the stress resistance of human cells, slowing down aging. Finally, their ability to survive in extreme conditions can make them available in outer space. Now I will cover the current status of tardigrades. There are about 1,100 described species of tardigrades, but many more are likely to be discovered, especially in non-terrestrial environments. It is also very interesting how they can survive after being completely dried out, frozen to 0 degrees Fahrenheit, heated at 300 plus degrees Fahrenheit, and may even be able to withstand the vacuum of outer space. Next, tardigrades have a few potential medical uses. Researchers are developing protein-based compounds that could stop bleeding and cell death and traumatic injuries. Secondly, they are researching how to slow down the body's metabolic processes after a traumatic injury, which could prevent damage from a stroke or a heart attack. Third, their proteins could improve insulin, which can clump up when exposed to heat or when it is shaken up too much, which can make medicine less effective. Finally, these may be able to stabilize pharmaceuticals. Here are all the resources we used during this presentation, and thank you so much for your time.